Hi students, welcome back to one more session of bioinformatics. In this session, we are going to study about sequence analysis. Of course, in the class, you have done various examples of how to explore sequences from the biological databases. So now we are just going to learn into I'm just going to explain to you what is sequence analysis. As we know, you have a database. It can be a protein database or it can be a nucleic acid database. You usually take the data, that is nucleic acid sequence, as well as the protein sequence from the nucleotide or the protein database, and that is referred to as database retrieval. After that, you can either take that particular nucleotide sequence and you can check either uh, what is its restriction enzyme mapping. That is, you can decide where all this DNA sequence can be cut, where all this DNA can be folded, or you can check for its coding sequences, or you can check into like what are the amino acids probably that can be formed from these coding regions. And you can if you're going to take the proteins, okay, you can get protein sequence analysis can be done. You can also do a prediction of the function. And if you go to take nucleic acid sequence, that can also come from different other things, like you can do sequencing project. That is, if you take a particular organism, you can, if, in fact, you can sequence the whole genome of that particular organism. And sometimes you can also take this nucleic acid sequence and design some primers. What are primers? Now, you know about the PCR. The PCR is a machine which is used to prepare different copies of DNA sequence. So for that you need some short sequences for to design, that is called a primer. So for the designing of primer, you can use nucleic acid sequences, or you can use it for various other analysis like BLAST and all that. And you can go to check the evolutionary relationship of different microbes, as well as different macroscopic or even human beings also. Like, you can find out their evolutionary relationships also by using various bioinformatic tools. So we're going to talk about overall of the sequence analysis and what are the different applications of these bioinformatics in this session. We'll be dealing with nucleic acid sequences as well as protein sequences, and what are the different practical aspects of these sequences which have been retrieved from the biological databases. That's what we are going to analyze in the coming sessions. So let's have an overview of the sequence analysis, which I had explained earlier. I'm just showing a pictorial representation of that. You can get data, nucleic acid data from a database, and that can be either used to find out how many restriction enzymes can cut that enzyme, or what is the 3D, as well as to find out the folding pattern of that particular nucleic acid sequence, what are the coding regions which are been from it, the translation to amino acids. Then we have the protein sequences. The protein sequence analysis can be done, predict the function can be done. And if you, as we mentioned earlier, you can take the sequence from a particular project. You can take a sequence, you can design a primer for it. You can take a nucleic acid, you can check what is the sequence. You can take that and you can check the similarity. Sometimes two nucleotide sequences similarity can be done, checked, or multiple sequence alignment. That is, more than two sequences can be analyzed for their similarity using a multiple sequence alignment technique. And when you're using, now I think you remember this, the database similarity checking is being done, and in that is mainly by blasting. Okay, so when two sequences are being checked or are being analyzed, or the sequence similarity between two sequences are being analyzed, we call it as a pairwise sequence comparison. And when we check the sequence similarity between multiple sequences, you use it as a multiple sequence alignment. Now, this multiple sequence alignment can be used either to find out the evolutionary relationship, that is, how distant or how are they related with other organisms, or you can check the motifs or patterns which have been present in some nucleic acids as well as in proteins, and to predict the structure as well as for the structural analysis. So in the coming sessions, we are going to find an overall of, not in a very elaborate way, but in a brief manner, we would be analyzing nucleic acids analysis and the sequence analysis and the basics of that. The most common software that we used for sequence analysis is the database similarity searching. Okay, and among that you have different versions that we have. Among that, BLAST is the most common one. Okay, here it's very popular, and you can see that you have done it. Then we also have the FASTA, which is also another one. You can have some sequences which are doing both BLAST as well as FASTA. So let's go into the details of it. I will be going into detail of that. Now, what is FASTA? It is a file format in which the DNA sequence is being formed, and it is mainly a simplest format. And you can see that when you write a FASTA format, you might have noticed in every nucleotide sequence as well as in all the protein sequences, when we are trying to retrieve it from the database, you can see that it is in FASTA format. Now, what is a FASTA format? The FASTA format is in such a way that the nucleotide sequence or the base sequence is written in a format like this. That is, you will have an icon like this. You will give the details of the file name, and you put enter, and you put all the nucleotide sequences in an order, one after the other. No spaces, no gaps are there in between. You just put enter. This greater than sign has been put. You put the file name, and then you put the nucleotide sequences. Now the same FASTA format can be used also for the protein sequences also. Here now this is what? Thymine, cytosine, cytosine. The only difference in a FASTA format for a protein sequence and a nucleotide sequence is that it will be the amino acids which are being given in the FASTA format of a protein. And in the FASTA format of a nucleic acid, it will be the nucleotides which are being given here. The nucleotide bases will be given over here. Now, you might have a doubt that I had mentioned FASTA earlier along with two different things. That is, among the blasting also we had mentioned FASTA. Is this FASTA and this here the same? This is a format. Now, the one which I explained here is a FASTA format. That is the sequence, the format at which a DNA sequence is being uploaded. FASTA, we will be dealing with that. It's in fact a pairwise alignment software. And in that, the DNA sequences or the nucleotide sequences were being given in a FASTA format, or the format in which the sequences were being given is this one. And so we consider this as a FASTA format of the particular file. Now, the FASTA format of a particular sequence should contain it should start always with a greater than sign followed by the sequence name, and the second line to end, it should have the sequence itself. The second line, this is the second line. It should, from the second line till the end, it should be the sequence. And in between of that, there should be no numbers, no spaces, and the sequences can either be in the upper or in the lower case. So this is about the FASTA format. Now, the file formats, among the file formats, you can see that most of the sequence analysis programs will take input sequences in this FASTA format only. And read sequence is a web-based utility which usually converts any file format into FASTA format. And many more programs will accept multiple file formats as the inputs. So the most common file format of a sequence, let it be a nucleotide as well as a protein base, it is a FASTA format. Now we're going to talk about sequence alignment. What is a sequence alignment? If you get a particular sequence of two different organisms, you might need to find out the relationship between those two sequences. For that, what you do, you will keep them together. You align them, that's what. You make them stay close together, and you check a residue to a residue correspondence are being analyzed. Now here in this particular case, you are aligning this particular sequence and this particular sequence, and you find that there are some interactions, that is, they are being found to be similar in some places. Here C and G is not the same, but T and T is, so there is a probable alignment over here. And T and T is a, though there is a gapped area where it has been found to be different. When you do the sequence alignment, or when you place the sequences close to each other, you can see that at certain areas they will be found to be similar. And you here, what you do, you do a precise operators for alignment. What do? You do matching, as well as you find out the gaps also. And there would be also a quantitative scoring system for these matches and the gaps. And the systematic search of all the possible alignments is being done when you do a sequence alignment. And certain algorithms are also being used for the optimal alignment. Now, when we get a particular sequence, we just input the sequence, and it is the software or the algorithm of the software which aligns these two sequences, and they will check the matches and the gaps. What are the possible similarities? What are the possible alignments? That is all been analyzed by various algorithms. Now, if you do a pairwise alignment is nothing but you take two sequences and then check whether they are having possible alignment frequencies, and they are being obtained by inserting gaps such that the resulting sequences have the same length, and where each pair of the residues represents a homologous composition. So you are just checking, aligning them both in such a way and checking whether how many gaps are there, where all the sequences are being found to be the same. So if you do alignment for two sequences, you call it as a pairwise alignment. And this is a case of sequence A and sequence B. You are aligning. You see, look here. When you have C, after C, there are certain sequences present here. Now, C, G, G, A. Okay, C, G, G is not there. So, or A is also not there. So, I put some gap. That is also a pairwise alignment where I put a gap in between the sequence. And sometimes what happens, you can see here T, T, A, A, C, T. That here T and A is being found, but C and T is different. But I did an alignment like this. Such an algorithm based alignment is being done by the software when you do a pairwise. That is, every nucleotide sequence when you have to analyze it with another sequence, alignment is being done with the known database, either putting gaps in between the sequences which have been found to be dissimilar, or by matching the homologous sequences. Now here you can see that here there is a match. Here there is a mismatch. Here there is a deletion gap. Here there is a insertion gap. Like that, when you analyze a pairwise alignment, you find different things. Now, similarly, you can also do a multiple sequence alignment. What is a multiple sequence alignment? That is, you take more than two sequences, okay, and the n sequences are being aligned simultaneously, where n is greater than 2. Now, this can be both nucleic acids as well as amino acids. And to construct a multiple sequence alignment, one may have to introduce gap into the sequences at positions where there are no gaps in the corresponding pairwise alignment. And multiple alignments typically contain more gaps than any given pair of aligned sequences. So, just like in the paired alignment, you see sometimes you might have to put gaps. Similarly, in multiple sequence alignments, you have many sequences more than 2, and sometimes more gaps will be put into the multiple sequence alignment. Now, this is a example of a multiple sequence alignment where 6 sequences are being aligned. And there are regions where they are being found to be similar. You can see that. See, here T is being found to be the same. GCA that has been found to be the same. So when you, if now they have some mismatches or it is not the same, they will put a gap over here. And this is almost, but there are other regions also. Now here, TGA, GTA, it's different, but they have aligned it here in such a way that they become homologous. And the homologous regions are also being identified. And after the process of alignment, you can see that when these sequences have been aligned, you can see sometimes there will be gapped areas when there are, as we mentioned in the case of pairwise alignment itself, here some gaps are also being produced when the sequences are not being the same. So I guess multiple sequence alignment is, you got a basic outline of it. Thank you for now.